Friedrich Mischner laboratory, the Max Planck, and we will talk about the Novo protein design.
Yeah, thank you. And thanks to Fergus for introducing the concepts of the Novo design and scaffold hopping, because I will speak about the same just for proteins. So the title is The Novo Protein Design of Potent Hematopoietic Agents. So at the beginning, I will start by introducing the motivation of why do we need active superfolding protein molecules, and then I will speak about two different strategies that we have tried to follow in order to achieve this. So to begin with, just to explain the current problems that are still standing with protein therapeutics, is that proteins are very complex molecules, and this molecular complexity translates into configurational complexity. And while we only need one configuration in all of this configuration landscape, which is the native folded active form, this active form still partitions to a lot of other inactive forms, and it's heavily distracted. And this partitioning into these other non-active forms is primarily governed by three different parameters. And these parameters are the folding thermodynamics, the folding kinetics, and the solvation-free energy. And the problems generally that result from this and the ways we try to mitigate it presently is that we have a lot of conformational heterogeneity for the protein molecules that are intended for therapeutic purposes, and also the excessive presence of post-translational modifications that are essential for folding of these molecules necessitate complex expression systems that are very costly, and also a lot of tangential protein engineering and also the formulation costs that are downstream of this process. For example, even things like pegylation, FC fusions, and extenuations, these are all adding to the complexity of the molecules but not simplifying them. But the three parameters that I explained, the solubility, the folding rate, and the folding thermodynamics, are themselves functions of three other parameters that are perfectly controllable. And these are the topology represented by the contact order, the size of the protein represented by the number of residues, and the sequence composition, the composition of the protein represented by its sequence. So in light of this, and in light of all of the theoretical and experimental studies that have demonstrated this, our assumption here is that if we have an active epitope, if this epitope's steric and electrostatic fields and also the associated dynamics are preserved, then every other part of the protein that is just contributing the scaffolding is fully exchangeable. And I will explain how can we optimize these parameters now using two different strategies. First is essentially scaffold hopping. So we disembody, if we have an active biologic, an active molecule, we just disembody the active epitope and try to mount it onto a geometrically accommodating scaffold. And the other approach is about rewiring the whole protein from behind the active site in order to make it much simpler, decrease its contact order, and decrease its topological complexity. So we started the experiments actually beginning of last year, and we chose a proof of concept as a family of molecules named the cytokines, because they are really important and they are central to a lot of diseases and disorders, as they are really at the crossroads of hematopoietic and immune cell maturation. So these agents are responsible for getting the bone marrow stem cells to differentiate, proliferate, and mobilize to the bloodstream to provide a host of functions of immune and hematopoietic nature. And every one molecule of these are deployed heavily in clinic to achieve this. It's only, their problems is that they are very unstable, they are heavily post-translationally modified, and they don't have really the optimal pharmacokinetics. So we have been working now on a few cytokines, but I will only explain the GCSF, the granulocyte colony stimulating factor, which is responsible for making neutrophils, as this cell type here. But first, I will introduce you a little bit more detail about the strategy. So this is the GCSF, and it has two binding sites to its receptor molecules. The most important site is called site number 2, which we dedicated all of the design work to here. So describing the first strategy, which is the topological refactoring strategy, which is aiming at topological simplification. Essentially, if we need to do this, we need to look at the loops, because if we can control the loops and design them de novo, we can achieve so much with this. So it has been already known historically in the literature that loops can, as we lengthen loops and we're putting unstructured loops, we can heavily disturb the folding thermodynamics and folding kinetics. But also, even in a very stable helical bundle structure, if we just take one of these loops and do saturation mutagenesis to just a single residue there, we can spend almost 4 kilocalories per mole in free energy change of folding, which is more or less the order of magnitude of folding a protein of this size. But more important and more strategically about loops, if we have an arrangement of secondary structures in space, that is a protein architecture. If we have this architecture, the combinatorics of connecting these secondary structures in space can explode in a factorial term. So while this is a statistic of the number of structures in the PDB and the number of secondary structures in each of these domains, and this is plotted against the linear scale of the number of structures, we can see a logarithmic scale, log base 10, of all of the possible structures given the same number of secondary structure arrangements, just by changing loop connectivity. So it's really, if we can control the loop design, we can, especially the Novo loop design, we can tap into an almost infinite resource of topologies. So to begin with, this is how the granulocyte colony stimulating factor looks like, and the aim was to eliminate unnecessary secondary structures in the back and also only keep the binding site, which is essentially here. And for that, we ditched 2 long loops and one helix and replaced them by 2 short loops. And by this, we can really achieve the theoretical minimum for a 4-helix bundle, theoretical minimum of the contact order for a 4-helix bundle. And I need to stress that, emphasize that the contact order is directly proportional to the misfolding rate. So the lower your contact order, the less misfolding propensity you have for a protein. And on top of this, we did a lot of other mutations. We added mutations at the core to fully repack the core again to make it more stable. And every other surface residue that is not involved in receptor binding, we just hydrophilized it in order to enhance the solubility of this protein. The other strategy, which is the scaffold hopping, is primarily focused on just dislodging the active site residues and trying to mount them onto, this is essentially a tertiary structural motif. We try to mount it on geometrically accommodating scaffolds. So we developed a particular method here for alignment-free geometrical matching of tertiary structural scaffolds, and these are the top 6 hits. You see they come in different flavors. And we tried only 2 of them. We tried, I guess, this one and that one. One of them is a coiled coil, another is a bacterial 4-helix bundle of an unknown function. So again, as we did with the previous design, we also hydrophilized the non-involved residues, redesigned some core when necessary, and also added some loops when necessary. So to get a quick recap on the experimental results that we have, first with the topological refactoring strategy, so redesigning loops de novo, we see compared to the natural GCSF, which melts at around 55 centigrades. So these are melting curves, using circular dichroism. Both are showing typical helical property, and while the wild type melts at 55 centigrades, the design doesn't melt at all, and the helicity is fully reversible upon melting to 100 degrees and cooling it back. For the other 2 designs that follow the scaffold hopping, the topological, the rescaffolding strategy, one doesn't melt and looks somehow reversible, and the other one melts at around 70. So at least both were still more stable than the wild type protein. We started testing now for the activity in something called the NFS60 cell line, which is a cell line derived from mice. It shows only proliferation in response to GCSF, and for the loop redesign molecules, we get a best result of half a nanomolar EC50. That's still 1.5 orders of magnitude less active than the wild type, but it's from the first hit of design, so we think this is very good because I need to say here that we took 4 designs of that strategy and 4 designs of the other strategy to the lab, so we only tested 8 molecules. And the 2 most active molecules here are shown. So this is the negative control and this is positive control of cells, hematopoietic stem cells obtained from healthy human donor bone marrow. And this is a time-lapse imaging of how they proliferate on the different treatments. This is untreated, this is treated with the wild type, and these are treated with 2 different designs. So we see a clear proliferative potential. Also, we see this with the designs from the other strategy, but albeit at lower EC50s, worse EC50s, so we get around 20-something nanomolar for these. Now, after checking that, we now go ahead and check for the differentiation. So how specific are these cells turning into neutrophils? So this is cell sorting results. We're looking for essentially double positives on, so whatever that is, how much cell counts we're getting at the upper right quadrant. And these are double positives on different markers that are labeled in the cell sorting by antibodies. To see how specific these 2 designs are, for example, compared to the GCSF. And these were the most specific designs. We also look at normal bright field microscopy. We see how the GCSF is making these segmented nuclei of neutrophils, classical, really textbook looking like, and also BOSCAR3 and BOSCAR4, which are very good designs, are showing exactly the same, which is at least these. Yeah, even maybe a bit bad, but disoherto is showing a little. So that looks really good. Now, we look at the major degradation pathway of these, of the wild type, which is the neutrophil elastase. It's when GCSF induces neutrophils as a negative feedback loop release neutrophil elastase, which degrades specifically the GCSF. Now, this is, you see BOSCAR4 here on the lanes is just getting its tag degraded, but it's very stable compared to the wild type, which is totally digested after 5 minutes. We see the same with one other design, not MEVN. MEVN gets degraded, but at least disoherto is still stable somehow, while the wild type is completely degraded throughout the protease treatment. We solved the structure of 2 molecules so far by using NMR, and we get, I can claim atomic accuracy here, with worst agreement between design and experimental coordinates at around 3.25 angstroms. And lastly, we have some preliminary data, but I wouldn't speak about these with confidence because we need to do some controlling experiments as well. So these are results of quantification on the y-axis of the neutrophils in a zebrafish cell line that has fluorescent neutrophils. And this is treatment with GCSF uninjected and 2 designs, and also here with BOSCAR4. What we can see consistently is the delayed action. So on the first day of treatment, GCSF is active, but our design is not active enough. But on the second day of treatment, we really get a shoot of at least a spike of neutropoieses that looks very specific. Also, we did, lastly, this is the last slide, we did some preliminary experiment with mice, but again, the numbers of animals here is not so big, so I wouldn't assert it yet. But again, we expect the neutrophil population to be around here. This is untreated, wild type after 3 days of treatment. And in fact, after 3 days of treatment, we see something consistent with what we see in the fish, is that it has overall lower neutrophil counts than we see with BOSCAR3 and BOSCAR4. We also quantify the neutrophils that are mature in the bone marrow, because we noticed in the fish that the neutropoiesis is so excessive when induced by the design that a lot of it stays in the bone marrow, and it's clearly and significantly higher than the untreated. So in conclusion, I just showed that how can we make novel post-translational modifications, free, stable, and highly solid molecules. And this is a very fast in silico to in vivo discovery and preclinical validation that we have demonstrated, because, I mean, in the computer, this whole thing didn't take more than 3 months of computing. And we got to the best results of single-digit nanomolar activity, and now we are still working on the in vivo characterization. But at least we can tell that we have pharmacokinetic superiority for the designs. Lastly, there is a poster, if you need more details, there is a poster A75. And by this, I would like to thank everybody that contributed to this, Patrick Mueller, my current supervisor, Bertha, Murray, and Julia, who are group leaders that are collaborating with us on this, Andre, who's my previous boss, and the work is still going in his lab. I did 1 year of this whole work under his supervision, and then Catherine, Christina, and Laura for also participating. Yeah, this is a backup slide. Thank you.
Yeah, thanks for a great talk. When in your scaffold, what do you call it, jumping or something?
Hopping, yeah.
Hopping, right. It was a little difficult to see, but it actually looked like that your new scaffold is actually, geometrically similar to the original scaffold. Is that right? So you have helix, you have a helix bundle in a similar conformation. Is that not right?
So you cannot really tell by the look because it's just so these residues are lying on two helices, right? And they are facing the solvent. This tertiary structure motif should match a span of around 10 and 10 amino acids on two parallel helices, and all of these have that. You cannot tell by the eye from the overall fold.
Right. I mean, so what I meant to say, compared from your wild type cytokine to the new scaffold, you always have the two parallel helices that are in similar conformations, right? So
that's right.
Okay, so it's actually a quite similar protein.
If you look at the C alpha C beta vectors, this is a constraint in this particular. Yeah.
Okay, makes sense. Thanks.
Yeah.
There's another question on the side.
Yeah. Perhaps I missed it, but what tool did you use to actually design the sequence? Like when you were redesigning the core and trying to do the novel loops?
Yeah, so we used a combination of tools. Some of the stuff is published, some other is not yet to be published, and I have a guess. This is one way of doing it. So this is, we use this to judge some, so this is a method we developed ourselves where we're trying to see the enthalpy contacts of one residue versus all of the other mutants, right? So we're looking at the energy transfer of an excited residue to all of the rest of the protein, and you hope that the ones that are very strongly enthalpically coupled are actually stabilizing the protein. Yeah, because in a design problem, you need to maximize the enthalpies, right? The second one, which is the crucial one, is the loop design, and we use a special type of replica exchange method. We also developed that ourselves, and you can see here, this is an example of two loops being simultaneously designed. So what we do here is that we essentially, let's say you have two loops that you need to design here across these two helices. You would, of course, clear the structure of this and have the two distances and fill them, let's say, depending on the distance, whether you need to fill these by 3 or 4 amino acids. So you would take, normally we take the top 8 amino acids with loop probensity from, just by their identities, and do all of the combinatorics. So if you have 3 residue gap, then you will take 8 to the power of 3. Yeah? And if you have 4 residue gap, you'll take 8 to the power of 4. And our simulations are so fast that we can try all of the combinatorics, but we do it in a greedy fashion. So we do the simulations for one loop first, and then the second loop, and simulate the other as a polyglycine. And then we take the top portions of both and do all of the combinatorics. Let's say top 50, and you get 2,500 or something like this, yeah? And you do all the simulations, and there is a factor score that we have developed as well. It tells you about the conformational heterogeneity of these replica exchange simulations. It works with every project. We have other projects that I'm not presenting here, but it works.
Thank you.
Okay. If you have no questions, we thank you for your presentation, and we go to the next speaker. Actually, we were expecting